In this lab, we're shifting from herbaceous plant structure to woody plants, the stems and roots that increase in thickness over time. Instead of preparing slides yourself, you'll be working with photos of microscope slides and labeled images, just like you did in the roots lab. The goal here is to learn how to recognize tissues, understand where they come from, and connect structure to function in woody stems and roots. These are the specific skills and concepts you should be able to handle by the end of this lab. You'll start by getting familiar with the key features of woody twigs, which are useful for identifying plants, even when leaves aren't present. You'll also learn what abscission is and why it matters for woody plant survival, especially in seasonal environments. Then we'll move inside the plant and compare the internal structure of woody stems and woody roots, focusing on the tissues you should recognize in each. Finally, you'll learn how to tell the difference between softwoods and hardwoods, between springwood and summerwood, and how annual rings form. You'll also practice identifying ring porous, diffuse porous, and non-porous wood types using microscope images. All of this ties back to understanding how woody plants grow, survive, and function over time. Let's start with how plants grow. Plants grow in specific regions called meristems, which are areas of active cell division. We can actually classify meristems in two different ways, by origin and by location. By origin, we talk about primary meristems, which are responsible for increasing the length of the plant, and secondary meristems, which are responsible for increasing the thickness or girth of stems and roots. By location, we use different names. Apical meristems are found at the tips of shoots and roots and drive primary growth. Lateral meristems run along the sides of stems and roots and are responsible for secondary growth. This is where the vascular cambium and cork cambium fit in. Intercalary meristems are found at the bases of leaves or internodes, especially in grasses, and they allow certain plants to keep growing even after being grazed or cut back. So primary versus secondary tells us what kind of growth is happening, while apical, lateral, and intercalary tell us where that growth is happening. There are two main secondary meristems we care about in woody plants, the vascular cambium and the cork cambium. These are rings or layers of dividing cells that continuously add new tissue. As they divide, they produce new cells both inward and outward, which is how stems and roots increase in diameter year after year. Every stem and root starts out herbaceous, meaning soft and green. In woody plants, older parts of these organs change. The epidermis gets replaced by cork, and the plant produces large amounts of secondary xylem, what we usually just call wood. This makes the plant stronger and better protected. The vascular cambium is the tissue that really drives wood formation. It makes secondary xylem on the inside, that's the wood, and secondary phloem on the outside, which becomes part of what we call bark. This process repeats year after year and is why trees get wider as they age. The cork cambium makes the plant's protective outer covering. It produces cork cells to the outside and phloderm to the inside. Along with the cork cambium itself, these tissues make up the periderm, which replaces the epidermis and woody parts of the plant. In everyday language, we call the outside of a tree bark, but botanically, bark has a specific meaning and includes all the tissues outside the vascular cambium, not just the cork you see on the surface. So bark is actually a combination of several different tissues. Becoming woody gives plants some big advantages. Wood provides strong structural support, letting plants grow taller and live longer. The cork and bark protect the inner tissues from water loss, injury, and pathogens, so secondary growth is a huge evolutionary advantage for trees and shrubs. In this lab, you'll look at both the outside and inside of woody plants. You'll examine twig features, study leaf abscission, compare woody stems and roots, and learn to recognize different types of wood based on their microscopic structure. The big goal here is to understand how woody plants grow in thickness, how tissues like wood and bark are formed, and how structure connects to function. By the end of this lab, you should be much more comfortable looking at a woody stem or root, whether in a photo or under a microscope, and knowing what you're actually looking at.